Hi everyone, we're just going to wait a few minutes for some more people to join and then we will get started. Hi everyone, we're just going to wait for a few more to join. Hi everyone, we're just going to wait another minute or so longer for a few more people to join and then we'll get started. Okay, we'll get started. Good afternoon, everyone. Good morning, and welcome to our webinar, Unlocking the Future of Skin Biopsy, Harpera Microbiopsy Punch. We're thrilled to have you join us today as we explore this innovative technology. Before we begin, just a quick note on webinar etiquette. So your microphones will remain muted throughout the session to ensure a smooth experience. Cameras are turned off automatically, so no need to adjust your settings. And if you have any questions, feel free to drop them in the Q&A section at any time, and we will address them during our dedicated Q&A session at the end. Now, I'm pleased to introduce our speaker today, Florian Lapierre, Product Director of the Microsampling Business Unit here at Trajan Scientific and Medical. Florian will guide us through the groundbreaking capabilities of Trajan's new Harpera Microbiopsy Punch. Florian, over to you.
Thank you, Camilla. And thank you everyone for attending this webinar. So today I'm going to talk to you about the Harpera Microbiopsy Punch, a minimally invasive microbiopsy alternative to traditional biopsy for monitoring skin condition. So as Camilla said, my name is Dr. Florian Lapierre. I am Product Director at Trajan Scientific and Medical within the Microsampling Business Unit. So first of all, maybe a bit of an history of where this microbiopsy technology comes from. It actually was invented by two professors in dermatology at the Dermatology Research Center from the University of Queensland. There was Professor Harpera and Professor Peter Sawyer, who saw actually an opportunity for microbiopsy in monitoring the condition of skin condition and in particular for skin cancer. And their idea behind the microbiopsy technology was to develop a tool that will allow the more frequent monitoring of skin condition to lead to early detection of those conditions. Also to provide a tool that enable a low risk and a minimally invasive procedure to promote the patient compliance during the sampling event. And also a tool that can enable a decentralized and also an economical procedure that makes the sampling accessible for all, not only for adult, but also for pediatric or for elderly, also for cosmetic locations or sensitive area. And last point also, it was to develop this microbiopsy in order to get a microsample that is compatible with molecular biomarker for concrete results. So since 2022, the Trajan has had an exclusive license obtained by the University of Queensland in order to commercialize this technology. And now we are commercializing actually the Harpera skin microbiopsy punch as an FDA registered for marketing in the US. And the Harpera is, the Harpera microbiopsy punch, the name of the product, is a manual surgical device intended to enable the collection of a specimen from the cutaneous skin surface by a healthcare professional. So as I said, it is registered now in FDA, through the FDA for marketing in the US. And outside of the US, the Harpera is made available as an investigational use only product for use in performance studies, in clinical studies. So as I said, the Harpera device is a single use, it's manual, it's handheld, it's a disposable microbiopsy punch. So typically, basically very similar to a standard biopsy punch, except that it will collect a microbiopsy instead of a biopsy. And it's intended to achieve a controlled skin culture in order to obtain a specimen. It is manufactured under the ISO 13485, and each device is supplied individually packaged in a Tyvek pouch and sterile through gamma irradiation. Also, a quick note on the timeline for regulatory submission and registration. So as I said, today, as of March 2025, the Harpera has been registered with the FDA as a Class 1 medical device. And now we are proceeding with the submission of the EU technical file in order to get the CE marking for Harpera as a Class 2A medical device around 2026. And after that, we will initiate a worldwide registration in key markets, depending on the market uptake. So whether in Brazil, in Australia, UK, Japan, and so forth. During this time, I didn't mention, but during this time, the Harpera will remain available also as an investigational use-only device for research studies and clinical investigation worldwide. So, what is the core IP that Trajan has licensed? It is the microbiopsy collector, basically. So this microbiopsy collector is made of 3 layers of stainless steel that has been laser cut through a precision laser cut, and then folded in a way like an origami, in a way that it can penetrate smoothly the skin without enduring any pain to the patient. And there is the roughness and the shape of the microbiopsy collector enable the grabbing of skin cells from the epidermis up to the dermis and to retrieve them outside of the body. You can see a close-up picture actually here of the microbiopsy collector. So this is the element that will penetrate the skin tissue. And this, you see the microbiopsy collector has a length of 1.2 millimeter long. So that means that it will not go as deep to touch the nerves or even the capillary blood. So that will lead to minimal pain and also minimal bleeding. And then the chamber, but also the outside layer of each stainless steel can be. This is where the skin cell will be grabbed on to be collected. And people have observed a different type of skin cells from the stratum corneum, the epidermis, up to the dermis being collected inside the microbiopsy specimen. Typically, to give you an order of idea, a microbiopsy specimen will generate about 1,000 of live cells with nuclei, for instance. So the key feature of Harpera, as I said, is the microbiopsy collector from high precision laser cut stainless steel. This is the core technology that is integrated inside the Harpera microbiopsy punch. And then we added a safety cap and a safety insert for enhanced protection of the end user that will utilize the punch onto a patient, and also an ergonomic handle for less handling. You will see in the video later on that in just one click, the sample is being collected. And last point, it's also a consistent and controlled punch actuation for reliable sample collection. That means that the spring mechanism with the plunger inside will always generate a consistent punch actuation, and it will not be dependent on the expertise of the dermatologist or the feel of the penetration depth through a punch biopsy. This is with the microbiopsy and the Harpera punch. All of this is controlled through the spring mechanism. In this slide, what you have to retain basically is that we are selling the device as a pack of 20 units inside a dispensing box. And today we are the price of the product of the 20 units is about $370 per box. And the product has a shelf life of 12 months. And this is the device, as you can see inside its individual Tyvek pouch. So let's have a quick look of the action of the Harpera for collecting a microbiopsy specimen. So after you teared off the Tyvek pouch, you have access to the device, and then you can remove the safety insert and the safety cap, and then pull the plunger in order to set the device. Then you have your dermatology patient, and you place the device vertically against the subject's skin surface. You press firmly the device onto it, and you click on the release lever. And that's it. After you pull off the device from the skin, and the skin specimen has been collected. So when we look at the clinical follow-up of a microbiopsy, what have we retained? First of all, there is no need for anesthesia or no pre-treatment of the area before the sampling collection. The second point also is that the puncture site is about a few hundred micrometer side. It's very much alike to a mosquito bite without the inflammatory reaction. From a patient perspective, the pain score is very low. It's about 1.5 out of 10 or on 180 volunteers. So it's there is a very good apprehension of the sampling event by the patient. And then post-sampling, there is a local erythema that is usually resolved within 24 hours. So that means that there is no need for any suturing, any post-sampling treatment, ointment, and so forth. And usually the skin specimen, the target site and the sampling site is invisible to the naked eye after 24 hours. That means that you can resample at the same area after almost 24 hours. So the potential benefit of using a microbiopsy punch versus a traditional biopsy procedure is, as I said, there is no body site preparation, no need for anesthesia and additional tools to prepare the skin area. It's minimally invasive and rapid in terms of the collection process. So for the patient, this is very much appreciated. It's portable and it can be used by any healthcare provider. That means that you don't need to rely on a surgeon or dermatology specialist in order to collect a skin specimen. Any kind of nurse or technician can perform the sampling event, the sampling procedure to collect a skin specimen. Next, there is no need for any post-sampling treatment on the target area after the microbiopsy procedure, and it also leads to a short healing period for the patient. So that means that with the Harpera, it's very much suitable for a topical sampling where you really want to collect at a specific target area, for example, within an AV or multiple microbiopsy specimen within an AV, next to an AV and far away from an AV. It is also suitable for additional sampling at one site. So when you want to, similar to what I just said, to collect multiple skin specimens to understand the heterogeneity of the skin. It is also suitable for frequent sampling over time. If one wants to perform longitudinal monitoring of the skin condition over time through different treatments and so forth, you can do that with an Harpera microbiopsy punch. And last point, it is also suitable for to collect skin on sensitive areas such as lesion on the face, on the scalp, in the armpit, anywhere on the body without by still being ethically acceptable and by having a very good apprehension by the dermatology patient. So Harpera really enables simple and minimally invasive biopsy with less pain. This is what you have to take away. So it really facilitates the recruitment to clinical studies. If you have to apply to ethics approval, etc., to collect multiple skin specimens, Harpera will facilitate the recruitment and the approbation by the IRB committee because it allows a rapid collection with a minimally invasive punch with minimal discomfort, even on cosmetic and sensitive areas. It's also economical from the clinical point of view because it eliminates all the complex processes associated with the skin biopsy, such as anesthesia during the sample collection, wound suturing, post-procedure, or to perform the sampling in a clinical setting, in a clean environment, and so forth. And last point also is that it's fit for purpose. It really meets the requirement for targeted, decentralized clinical studies that require more frequent and routine monitoring of skin condition, not only just collecting a skin specimen to collect the full area and then do a diagnosis or do a monitoring, to do an analysis of the skin area later. Here it's really more to collect multiple skin specimens throughout time and understanding what's going on to the skin of the person. And when we look actually at the trend and the need from dermatologists are actually seeking more and more of these minimally invasive tools. And some of them already exist, like tape stripping and brush swabbing, that really much collect the top surface layer, the cutaneous skin, the stratum corneum surface of the skin in a large area. Or some other are repurposing tools like like needles or scalpels in order to be less less painful or more minimally invasive. But the Harpera really fit for dermatologists and researchers who really struggle to consistently and accurately evaluate the skin condition in a minimally invasive manner. The design of the Harpera has been made to provide a virtually painless way to routinely and precisely collect a skin specimen for targeted biomarker analysis. So now, what happens after you have collected a skin specimen, and what are the different trends of what people are doing after they are collecting their first microbiopsy skin specimen? So first of all, what they are doing basically is they are opening the casing of the Harpera punch either by hand or using a pair of pliers or tweezers. And then they got access to the plunger and the microbiopsy collector, and therefore they can grab the backhand of the microbiopsy collector with a pair of tweezers or pliers and then pull it out to put it then into a microcentrifuge tube. Then the next step is to preserve that microbiopsy specimen. And in the most common way, some people are using the wet preservation by utilizing different stabilizers like RNA-later, where they dip into the microbiopsy collector into RNA-later and keep it at room temperature up to 7 days, depending on the manufacturing instruction, or they snap frozen it. The other way also is to do the direct extraction of the microbiopsy, and then after the microbiopsy collector removal, do the snap frozen the extract basically. The other way also is to do the dry preservation. So that means after taking out the microbiopsy collector, performing directly a snap frozen of the microbiopsy collector without any liquid. And nowadays, actually, some of our group are testing out the room temperature. That means that similarly to a dry blood spot, what happens to the RNA or the DNA or the different biomarker if we just leave the Harpera as it is with the microbiopsy specimen inside the device to see whether there could be some good storage capability even at room temperature. So for now, this is under evaluation if it performs well or not. Then in terms of type of assay from the microbiopsy specimen, most of our customers are using the microbiopsy specimen for RNA or DNA type of analysis through either RT or qPCR for gene expression analysis, but also for RNA sequencing to measure the sequence of RNA. You can measure also DNA, so whether it's human DNA, some other people are measuring also the presence of parasite DNA inside the microbiopsy specimen. And nowadays, some more people are testing also other alternative way of the performance of the microbiopsy through proteomic analysis, metabolomic analysis, with Olink proteomic or LCMS technologies or ELISA. So I think that we are at the very start of what kind of microbiopsy specimen can provide to the community, and more researchers are evaluating its analytical performance. Some other people are also testing like the live cell tissue to do some live cell assay or cell culture directly from the microbiopsy specimen. So the takeaway of this is basically a microbiopsy collector will collect about 1,000 of skin cells. And so that means that if you want to implement the microbiopsy into your laboratory, we really recommend the laboratory to use molecular kits that are specifically designed for single cell type of analysis. So 1 to less than 1,000 of skin cells to recover RNA or DNA and so forth. And there are actually many off-the-shelf kits and endpoint instruments that are being used by our current customer, from Promega, Thermo Fisher, QIAGEN, and so forth. Typically, each collected microbiopsy specimen will generate about 1 to 10 nanogram of DNA/RNA. So that means that you cannot do everything with a microbiopsy, but you can do quite a lot from RNA-seq or gene expression analysis. And then also it will collect about 1 microgram of protein. For an RNA-seq application, right now the trend is that pulling 3 to 4 to 5 microbiopsy specimens together is more recommended in order to get sufficient quantity and quality RNA. We have a user guide that illustrates the various kits that are used and discussed in the literature. I think there's about 20-ish different publications that demonstrate the use of the microbiopsy in different dermatology applications. And the user guide illustrates some of the kits that were used in those publications. And also you can refer to us, or you can come to us and we can refer you to our partner laboratories if you want to have a support for your project. So here is an example, for example, of the different molecular kits that have been used in the literature. And you can see the main one, for example, the RNA-Z from MicroKit, is widely used, or the PicoPure from Thermo Fisher is also very much used. And then after, in terms of the endpoint instrument, it's mainly a qPCR kind of system, like the QuantStudio or the Biomark from Freedime is quite a good system to do some gene expression analysis. We are also working nowadays with different kit manufacturers like Promega or Biocrates in order to improve and set up and standardize the analytical workflow using their kits for utilizing microbiopsy specimen. So again, if you have some issue in generating sufficient yield, etc., please come back to us and we can refer to the product manager at Promega or Biocrates and so forth in order to help you and support you with the implementation of your workflow using microbiopsy specimen. Before I enter into some examples from the literature, here is some testimony from 3 different researchers but really showcase what the microbiopsy has enabled them to do. Basically, the microbiopsy and the Harpera microbiopsy punch really enable new possibilities that has been unexplored using classical methods. For example, Dr. Amar from the Technical University of Munich is using the Harpera microbiopsy punch for sampling babies and collecting actually multiple samples over time on babies without any issue from the mother or the IRB. And they are using these microbiopsy specimens to better understand the genes for studying the atopic dermatitis skin disease. Dr. Yamada from the Hull York Medical School, they are routinely extracting RNA from a single microbiopsy with real-time PCR or even RNA-seq. Now they do like gene expression analysis. They analyze up to 210 genes with their workflow, and now they are moving forward with proteomic analysis. Or Mengye Shao from Shiseido, they are also using the microbiopsy for whole transcriptomic analysis, and they do also some cell culturing using the microbiopsy collectors. So now let me show you some use case scenario of the use of Harpera into some application. And this is actually one of the first publications that was published back in 2013 by the inventor of the technology to demonstrate the advantage of microbiopsy punch or to overcome the limitation of standard biopsy. So what they did is really much taking different volunteers, like there was 20 healthy volunteers. They also taken some excised skin from different lesions, and they measured different storage conditions, different type of analysis. They measured the pain score, etc. And basically, what their result was that there was a very good apprehension of the skin sampling by the volunteer, very low pain scale. They did all the clinical evaluation, so to understand the puncture site, looking at the local erythema, understanding how much cells were captured inside the microbiopsy, and to do some live cells assay. How much yield in terms of DNA and RNA was recovered from a microbiopsy specimen in order to do either whole transcriptome amplification, but also to assess whether they could recover some housekeeping genes from the total RNA extracted. And all of this kind of worked well. So they moved then forward with the core of their application, which was related to skin cancer and the biomarker discovery of skin cancer. And so they took the traditional method where they usually use a dermatoscopy. They take a clinical image, they perform a shave biopsy in order to do a subjective histopathology diagnosis, and at the same time they performed the microbiopsy procedure by collecting up to more than 1,000 of skin collection from head, neck, trunk, limbs, from everywhere, and also, as you can see in this illustration, within the lesion, so multiple skin microbiopsy specimen within the lesion, but also next to the lesion and very far away from the lesion. And then they assessed through real-time PCR assay the different biomarkers and trying to identify whether known or novel biomarker of basal BCC and melanoma biomarker. And so this is really much. So this has been published several times in dermatology publication. And basically, what they wanted to showcase is that by using Harpera, it can enable a personalized dermato-oncology by addressing the issue of intra and intertumor heterogeneity, by identifying the molecular signature of target within lesion, etc. Everything is basically to the end goal of all their research is basically to avoid the use of invasive skin removal procedure prior validated biomarker are identified. So that means that they receive patients where they got 100 of different moles and nevis, etc., which could be all suspicious, then they will be able to collect some skin microbiopsy specimen in order to measure whether there is a presence of a validated marker before conducting a punch biopsy. So the microbiopsy collector is not, the Harpera microbiopsy punch is not here necessarily to replace the punch biopsy procedure. Then some other groups have also used the microbiopsy punch for more cosmeceutical application. By, for example, in this case, when they published, they measured the oxidative stress of live cells using confocal microscopy. And what is really good in this study is that they applied on the forearm of patients 6 different treatments on the forearm, and then microbiopsied them 5 times per treatment site. So that means that the patient has endured, in a way, 30 microbiopsy at a single time point. And what was their takeaway is that, first of all, the skin cells were viable and intact. They could demonstrate through confocal microscopy that the degree of oxidative stress, depending on the treatment that they applied, and therefore that microbiopsy is a viable approach to study cosmeceutical for skin interaction. So it can be applied for pharmaceutical or for diagnostic or for dermatology application, but also for cosmeceutical application, because it has the capacity for frequent sampling in a short period of time with various treatment sites without any local anesthesia, bleeding, pain, or scarring. So that means it reduces the risk of skin sampling to its minimum. Another group in Czech Republic, they're about actually to publish their study in the PLOS journal. And what they are focusing on is understanding the factor influencing the transmission of Leishmania parasite in different hosts. And so what they are doing basically is that they are infecting the hair of rodents by a sandfly that is infected by the parasite. So the hair of the rodent, you can see here the lesion of the rodent where the sandfly has bitten and infected the hair with the parasite, and then they can retake a microbiopsy at the lesion site and then in periphery and further away in order to understand the parasite load and how it spreads and distributes throughout the body and over time. And this is only possible through the use of a microbiopsy punch, because it really enables a repeat collection of multiple lesions in an animal model. And so that means also it enables them to do more things like precise modeling of the cutaneous Leishmania epidemiology. And now they are moving forward with their study to not only count the parasite load, but also to do some mass spectrometry-based approach for rapid identification of hosts and vectors in this context. Another case also, I think I've got two more left, is the use of the Harpera again for cutaneous Leishmania, but now in human participants. So that means that the Harpera has been used for quite several years now in India, in Africa, in Brazil, in order to understand the parasite load in a human host. Who have the different lesions sometimes or who are sometimes asymptomatic. And what they were capable to do with the microbiopsy punch is that they could collect at a different sampling site, and especially in the preferred spot for sandflies where they bite. So that means the arm, the neck, the lips, in the cosmetic areas where usually the people do not want to get a biopsy punch or a shave biopsy being taken from their body. So the result of their study is, first of all, there was a very good compliance from the volunteer to participate in the study, even the participants which were asymptomatic. So that means presenting no lesion or no scarring, they were okay to be collected a skin microbiopsy. And the pain score were again very low, about 1 out of 10 on 148 participants. There was also a very good retention of participants in this study due to the minimal inconvenience. And now they are moving forward with even people which have HIV and which are more prevalent to be a host of those parasites, and which are very difficult to get access and get their compliance to participate to a clinical study. The tissue were collected on several sampling sites, and as I said, yeah, even on cosmetically sensitive areas and lesions, and even on pediatric patients. So the impact is really for them, for this group, is to improve the standard of care of low and middle income country, to provide an early diagnosis, and also to enable the species of the parasite identification in order to provide the adequate treatment, which is very hard on the patient. So it's very important to provide the adequate treatment prior to scar formation or stigmatization and so forth. And all of this in the end goal to improve the disease control and the surveillance of cutaneous leishmania in these countries. A last study is the use of the Harpera punch for whole transcriptomics analysis in the early childhood with children who have atopic dermatitis. And so what they have done for the past few years now is that they are including microbiopsy into their studies in order to collect more skin specimens that they could not collect before, but also to monitor throughout time the skin condition at a specific target site and throughout the age of the baby, from the birth to 48 months old. So this study is about actually to be concluded, and the group is about to publish very soon their result. So the conclusion of this and the key takeaway that you need to take is that, first of all, the Harpera Microbiopsy Punch is a registered medical device as a Class 1 in the US since March 2025. In other regions, the Harpera Punch is distributed as an investigational use only device for use in clinical studies. The microbiopsy sampling procedure with Harpera is minimally invasive. It's virtually painless, enabling dermatologists new possibilities that have been unexplored using conventional sampling methods. The microbiopsy specimens have been analyzed with various analytical techniques, and now Trajan is working with molecular kit manufacturer to standardize analytical workflow with microbiopsy specimens. So don't hesitate to contact your local neoteric specialist to test out microbiopsy skin sampling. So whether Tracy, Libby, Joseph, or even you have here all our contact detail. So don't hesitate to take a screenshot of this. And contact also myself at flapierre@trajanscimed.com if you have any technical question or you want to do some collaboration project. Again, don't hesitate to visit our Neoterics website, and please click on the Harpera page if you want to get the latest update on the Harpera Punch. And I thank you very much for your attention. I think I'm happy to answer any question you have.
Thank you, Florian. Yeah, there have been a couple of questions, so thank you so much for submitting those. So the first one, it's sort of a two-part question. Can genomic and proteomic biomarker endpoints be evaluated from microbiopsies collected via the Harpera tool? And does the microbiopsy sample quality and quantity suffice?
So the Harpera just does the sampling. It does not do the analysis and so forth. So in terms of the claim that we are doing as part of the medical device, we take the Harpera punch as a punch biopsy. So there is no diagnosis, no analytical workflow linked to the Harpera punch. Now there is groups who are doing proteomic analysis and metabolomic analysis, even lipidomic analysis from microbiopsy specimen. We are working, for example, with the company Biocrative, who develop metabolomic kits, and they got some very good results with that regard. So don't hesitate to approach us, and we can put you in relation with this company, or just test it out by yourself if you want to do it yourself.
Thanks, Florian. Okay, and then the next question, what are all the skin sites and types that have been tested? Palmar skin, facial, scalp, gingival.
Oh, it's quite a lot. I think the whole body, the whole surface of the body of the people of patients have been tested mainly by the inventor of the technology. So whether it's even the hand, the palm of the feet, the back leg, the armpit, etc. Maybe one thing that was retained from all this sampling is that it should be airless. So if there is, for example, if you are collecting from the scalp, you should ensure that maybe there is a minimal air during the sampling during the sampling event, because it could affect how the microbiopsy will penetrate into the skin to collect the microbiopsy specimen. So you will have to try and test out maybe the sampling. But yes, the least air, the better the sampling event is.
Thanks, Florian. There is another one here as well. How to choose the right spot on mole to perform a single punch? How accurate can this single punch be when it comes to getting cancer biomarkers?
So I think this is maybe a bit out of my expertise. I think you may want to read the different publication from the inventor of the technology. But so the Harpera is precise to collect in a navy, but you will not be able to say, okay, I want to collect exactly at that location and then exactly at that location. So you will collect within the navy, and depending on how you place the Harpera, you will hit a place, and a different place is each time you are placing another Harpera into it. So it's not like you say, I want exactly this position at the micrometer size, let's say.
Thanks. Another one here. Do you know any laboratories who can process microbiopsy specimen for molecular analysis?
Yep. So there are several laboratories who are in Europe and in the US who are actually using who are analyzing microbiopsy specimen. So you can contact myself or our commercial team in order to direct you to the most relevant laboratory. However, keep in mind that if you know a laboratory who knows how to process low tissue input, so single cell type of analysis, if you want to do RNA-seq or gene expression analysis, if they know how to use a QIAGEN, RNA-seq, or the PicoPure from Thermo Fisher, then this laboratory should be able to work with a microbiopsy specimen. So, yeah.
Okay. Another one, thank you. Is the method of sampling important, i.e. angle of the punch, how to remove the punch, skin tension, in determining the quantity of cells that you collect?
So we have not seen any indication that the sampling, how can I say? Sorry. Follow the IFU, because the IFU indicates to press firmly on the device onto the skin in order before pressing the release lever. Now, stretching the skin like a dermatologist would do prior to a punch biopsy, or not stretching the skin, we have not seen any indication whether it will collect more or less skin. Normally, by pressing firmly, the device onto the skin will usually stretch the skin in order for the microbiopsy collector to penetrate it. Now, we also recommend that the microbiopsy punch is placed vertically against the skin, so not at an angle or something like this, because otherwise it will not be able to penetrate the skin. So that's why the pressing firmly is important in order to be vertically vertically aligned to the skin. This press firmly can be also subjective. So what indicates a good sampling is that there is a mark left on the body of the person. There is sometimes a minimal bleeding depending on where you are collecting from, but there will be always a tiny mark, like a mosquito bite kind of mark. If you do not have this kind of mark, no sample has been collected, and then you can collect another sample. Another thing also to take into consideration, and this is why we are advising an healthcare professional to use the device and not do a self-sampling, is that the person that click on the release lever should not touch the plunger that will move during the sampling event. So if you give that to a healthcare professional, usually they understand that there is the plunger and they should not put their finger on the plunger or ensuring that the plunger can move freely. While if you give the Harpera punch to a lay person, they may not be able to understand that, and therefore you don't know whether a good skin biopsy has been collected or not, because they may have touched the plunger during the sampling event.
Thank you. And then there is one more here. Again, it's sort of two parts. How long does it take to receive the devices in the US, and do you send free samples for evaluation?
Yep. So as of right now, you can purchase the Harpera and you will get it within few days. We have a stock in the US in Austin, I believe, in our Austin warehouse. So if you contact our commercial team to ask, for example, a quote for one box, you should get it within few days. If you would like to receive a free sample, I'm pretty sure this can be done. Usually with one box, you've got 20. It costs $370, so it's not that much. You get 20 units. And then with 20 units, you should be able to get started, at least with testing out with your methodology, with whether clinically it's acceptable and so forth. And it's not like out of budget, let's say.
Okay. Well, I think that's all the questions for now. So thank you, Florian, and thank you everyone for joining us today and for your insightful questions. We really do appreciate your time and your engagement. If you have any further questions, please do not hesitate to reach out. Wishing you all a great day. Goodbye.
Thank you.
Thank you.